What should we know about digestion?
Digestion is the mechanical and chemical alteration of food that allows the absorption and use of nutrients. Digestive system is involved in the processing and absorbing of the important nutrients. Digestive enzymes will break down the large nutrients into smaller fragments. Firstly, the digestion and absorption of carbohydrates. Carbohydrates can be classified into polysaccharide, disaccharide and monosaccharide. While monosaccharide is important for absorption, carbohydrates digestion involves actions of salivary and pancreatic enzymes, action of brush borders enzyme. Carbohydrate digestion begins in the mouth during mastication, through the action of salivary amylase. Salivary amylase breaks down starch. Starches and glycogen is digested continuously before stomach acids render the enzyme inactive. What about stomach? Only a small amount of digestion takes place over this period because the enzymatic content of saliva is not high. In the duodenum, pancreatic alpha amylase breaks down remaining complex carbohydrates. Brush border enzymes of the intestinal microvilli break polysaccharides into monosaccharides prior to absorption. Here are some examples of brush border enzymes acting on the disaccharides. Lactose is the main carbohydrate in milk. If the intestinal mucosa stops producing lactase, the person becomes lactose intolerant. The intestinal epithelium then absorbs the monosaccharides by facilitated diffusion and cotransport mechanisms. Both facilitated diffusion and cotransport mechanism involve a carrier protein. Now, spot the differences. Facilitated diffusion moves only one molecule through the plasma membrane while cotransport can move more than one. Facilitated diffusion does not require ATP while in cotransport the cell must expend ATP to preserve homeostasis. Facilitated diffusion does not take place if there is an opposing concentration gradient but cotransport do. Here comes the last stage of carbohydrate digestion. The simple sugars that are transported into the cell at its apical surface diffuse through the cytoplasm. They reach the interstitial fluid by facilitated diffusion across the basolateral surfaces. Lastly, these monosaccharides diffuse into villous capillaries for eventual transport to the liver. Next, lipid digestion and absorption. Triglycerides consist of 3 fatty acids attached to a glycerol. Lipid digestion involves linguolipase and pancreatic lipase. Linguolipase breaks down triglycerides in mouth and continues within the stomach. Only 20% lipids is digested when the chyme enters duodenum. Bile salts emulsify the lipid, providing better access for pancreatic lipase. Pancreatic lipase then breaks the triglycerides. These molecules are released and interact with bile salts to form micelles. When micelle contacts the intestinal epithelium, lipids diffuse across plasma membrane and enter cytoplasm. The intestinal cells synthesize new triglycerides. These triglycerides and other components then coated with proteins, producing chylomicrons. The intestinal cells secrete chylomicrons by exocytosis. The protein coating keeps the chylomicrons suspended in interstitial fluid. Most chylomicrons diffuse into intestinal lacteals, proceed along lymphatic vessels and through the thoracic duct. Lastly, they enter the bloodstream at the left subclavian vein.
Protein digestion and absorption. Protein have very complex structure, so protein digestion is both complex and time-consuming. Proteinating enzyme must access individual proteins, so the three-dimensional organization of food must first be disrupted. This step involves mechanical processing in the oral cavity through mastication and chemical processing in the stomach through the actions of hydrochloric acid. The strong acid in the stomach kills the pathogens and breaks down plant cell and animal connective tissue. Acid also disrupts tertiary and secondary protein structure, exposing peptide bond to the enzyme attack. The acidic content of the stomach also provides the proper environment for the activity of pepsin, the proteinating enzyme secreted in an inactive form by the chief cell of the stomach. Pepsin works effectively at a pH of 1.5 to 2. It breaks the peptide bond within a polypeptide change. When chyme enters the duodenum, enteropeptidase from the small intestine trigger the conversion of trypsinogen to trypsin. Both will increase to the pH of 7 and 8. Trypsin, chymotrypsin, and elastase are like pepsin in that they break specific peptide bond within a polypeptide. Carboxypeptidase short off the last amino acid of the polypeptide chain, no matter which amino acid are involved. Thus, while the other peptidases generate a variety of short peptides, carboxypeptidase produce free amino acid. The epithelial surface of small intestine contains several peptidases, notably dipeptidases. These enzymes break short peptide chains in the individual amino acid. The amino acid diffuse through the cell to its basal surface. There, they are released into the interstitial fluid by facilitated diffusion and co-transport. Once in the interstitial fluid, the amino acid diffuse into the intestinal capillaries for transport to the liver by the means of hepatic water vein. Water absorption. This is the intestinal lining. So, cells in the body cannot actively absorb or secrete water. All the movements of water across the lining of the digestive tract involve the passive flows of water down the osmotic gradients. Since osmotic movement is rapid, the interstitial fluid and the fluids in intestinal lumen always have the same osmotic concentration of solutes. As the nutrients and ion being absorbed by the intestinal epithelial cell, the solute concentration in the lumen decreases. Water moves into the surrounding tissue to maintain the osmotic equilibrium. Based on the diagram, a normal person takes in roughly 1 to 2 liters of dietary fluid every day. Another 6 to 7 liters of fluid is received by the small intestine daily as secretion from the salivary glands, stomach, pancreas, liver, and the small intestine itself. By the time the ingesta enters the large intestine, approximately 80% of fluid has been absorbed. Lastly, left with 150 milliliters of water in the feces. Ion absorption. Sodium ion. Sodium ion is the most abundant cation in fruits, absorbed by diffusion, co-transport, and active transport. Rate of the uptake is proportional to the concentration in the intestinal contents. The more sodium ions in the intestine, the more sodium ion is being reabsorbed, and the more the water is being absorbed. The rate can be increased by the presence of aldosterone. Calcium ion. Calcium is absorbed by active transport. Calcitron increases the rate of transport. For magnesium, iron, phosphate, and sulfate ion is actively transported into the body. On the other hand, potassium, chloride, iodine, bicarbonate, and nitric ions are all transported through the canal-mediated diffusion. Vitamin absorption. As we know, vitamin is being categorized into water-soluble vitamin and lipid-soluble vitamin. Water-soluble vitamin comprising the vitamin B, which includes the folate, thiamine, riboflavin, and so on until vitamin B12, and also vitamin C. They are easily absorbed by diffusion across the digestive epithelium except vitamin B12. Vitamin B12, due to its large size and charged natures, it must bound to the intrinsic factors, a glycoprotein secreted by the parenchymal cell of the stomach. This combination then absorbed through the active transport. While for lipid-soluble vitamin, it includes the vitamin A, B, E, and K. Fat-soluble vitamin absorbed from shells along with the fatty acid and monoglyceride into the lactears. Okay, this is a note. Taking the supplements of fat-soluble vitamin while anti-stomach or an low-fat diet is relatively ineffective as this vitamin requires a lipid for its absorption. Thank you.